Well, welcome everyone this afternoon. I'm Julia Bradsher. I'm the President and CEO here at HMRI, and I want to welcome you to our Researchers Roundtable. I'd like to extend a warm welcome to everybody for your participation in this event. We are very pleased to have our first full cohort of postdoctoral fellows from all over the world here with us today, 6 of whom are going to present their research this afternoon. Our postdoctoral program is designed to recruit the best talent, both nationally and internationally, to our neurosciences, cardiovascular, and neurovascular research programs. We believe that a diverse postdoctoral community has very positive effects on research and teaching and enhances the work environment by offering broadened perspectives and encouraging critical thinking. Our postdocs have really engaged in that over the past few months as they've begun. And I also want to thank the James G. Boswell Foundation and the Gustavus and Louise Pfeiffer Research Foundation for their support for this program. So now I'd like to introduce Dr. Anju Vasudevan, our HMRI Scientific Director of Angiogenesis and Brain Development, and also the Director of our Postdoctoral Fellowship Program. Dr. Vasudevan.
Thank you, Julia, and welcome everyone. I'm delighted to share a few words about the postdoctoral program. The postdoctoral team that we have with us today is very unique because this team formed during the worst pandemic in 100 years. Our fellows had to overcome a lot of difficulties to come to HMRI. They are obviously very brave and passionate about research, and we believe that there is nothing that they cannot accomplish. The recruitment is still ongoing. We have new fellows on the way, and we hope that this program will continue to grow. So what exactly are we looking for in this program? Well, we believe that scientific curiosity and talent is international, it's universal. And therefore, this program is open for people all around the world. And with respect to the recruitment itself, what we are really looking for is a positive attitude and endurance, because we know that good science takes a lot of patience and resilience. And so a genuine sense of scientific curiosity is more impressive than just plain intelligence. And with respect to today's event, it's truly a special one because usually in the roundtable we have one topic and one speaker. But today we will get to hear about a wide range of topics that will illustrate the broad spectrum of science that we have at HMRI. Our fellows will tell us a little bit about themselves, about the work that they're doing, and what they hope to achieve in this program. So without any further delay, I'd like to move the floor to our postdoctoral fellows, and we'll take all questions in the end. I'd like to welcome JK to start the session.
Thank you for starting us off, Anju. My name is Jung-eun Ko. I go by JK, and I'm a postdoctoral scholar in the neuroscience program here at HMRI. Today, I'm excited to share my project in using luminescence to detect Alzheimer's disease. To briefly introduce myself, I'm trained as a materials chemist to develop materials to improve human health. I received my PhD degree at UCLA under Professor Heather Maynard, where I developed materials to improve protein drugs such as insulin and growth factors. And for my postdoc here at HMRI, I'm working with Dr. Alfred Fonte to develop an assay to detect Alzheimer's disease. So Alzheimer's disease is a devastating disease, not only for the patients, but for their families as well. It's the 6th leading cause of death in the US and 3rd among the elderly after cardiovascular disease and cancer. And in Alzheimer's patients, in their brains, the the proteins called amyloid and tau start to accumulate over time. And in this study involving almost 500 patients, they estimated that the amyloid continuously accumulates inside the brain, but the brain compensates for the loss of function. So it takes 10 to 20 years until the patient even experiences any symptom of the disease. And this symptom themselves are very hard to distinguish from the natural aging process. And making matters worse, the current diagnosis of Alzheimer's disease requires either a very invasive collection of the sample from the spine or the expensive PET scans, both of which presents a significant financial and also mental barrier to getting tested. So if we can develop an easy, accessible, and affordable assay for Alzheimer's disease, that would allow the patients to prepare for their future while they're mentally capable. And hopefully in the near future, when we develop more effective drugs to manage the disease, then the patients can start to get treated early to mitigate the progression of the disease. Here at HMRI, the scientists have identified these molecules called dicarboxylic acids, or DCAs. And the levels of DCAs in urine can be used to tell if the person is healthy or has the Alzheimer's disease. So in healthy patients, the level of shorter DCAs is higher than the longer DCAs, but in the Alzheimer's patients, the trend is reversed, where the longer DCAs, their level is higher than the shorter DCAs. But the process that we currently use requires very complicated equipment, so it cannot be readily used in a clinical setting all over the world as we hope. So my project is to use luminescence to make this process easier to detect the DCAs in urine. So luminescence is used in various fields in analytical science, and it's most popularly known in forensic science, where a compound known as luminol reacts with the iron in the blood to give off a strong luminescence signal to show the traces of blood that's been left behind. So if luminescence can be used to catch numerous numbers of killers, can we use the luminescence to detect the traces of the silent killer Alzheimer's? And this is our strategy on using luminescence for the Alzheimer's assay, where the patients can submit their urine by mail. They don't even need to make a visit to the doctor's office. And once received in the clinic, the clinician can treat the urine sample containing these DCAs, shown as the colored wiggly lines, with the luminescent probe and visualize these DCAs using luminescent light. And I'm happy to report that the prototype system that I have developed does show that it's possible, this strategy is definitely feasible, and I'm able to distinguish the different DCAs and show them by luminescence. And I'm currently working on improving the sensitivity of the assay so that it can be adopted in the clinic. And through this postdoctoral experience, I will be able to grow into a scientist who can solve real-world problems. And for my further in my future career, I'll use this as a stepping stone to build a research career where I'll use my training in materials chemistry to address problems that will benefit not only patients afflicted by Alzheimer's disease, but also other disease that I'll tackle in the future. I'd like to conclude my talk by thanking everyone who's made this work possible. First off, I'd like to thank the neurosciences group at HMRI, I'd like to thank Dr. Bob Kloner, the interim director of the program, Dr. Alfred Fonte, my mentor for the program, and also Dr. Shanghong Arakaki, and Dr. Anju Vasudevan for directing this postdoctoral program. And I'd like to thank my mentor at Caltech, Dr. Bob Grubbs, and my collaborator, Dr. Mark Davis. I'd also like to thank HMRI for giving me this wonderful opportunity to contribute to the neuroscience program, and also the Kavli Nanoscience Institute for additional support. And I'd like to thank you all for your attention.
Hi, this is Dr. Pranay Goyal. I'm in the neurovascular research program at HMRI. I received my doctorate degree from the Department of Medicine at the University of Adelaide in Australia. I can give you a brief outline about my research interests. I like to study disease mechanisms using animal models, elucidating the mechanisms that lead up to the disease. By understanding disease biology, I can find molecular targets that can be used further for drug development. I like to create new preclinical models of the disease. Also, I like to develop biomarkers for disease detection and novel techniques to dissect disease etiology. Further, mental illness affects 1 billion people worldwide. This is a huge stigma associated with mental illness, and many people around the world are suffering in silence. Also, most of the treatments for psychiatric disorders today can only treat the symptoms, and there is no cure. Multiple factors can contribute to psychiatric disorders. These factors include genetic predisposition, developmental insult, brain injury, and aging. Together, these factors can influence the molecular, physiological, and structural characteristics of the brain. Environmental factors such as demands of life, loss of a loved one, or experience of abuse can also have a negative impact on brain health. Taken together, there are multiple variables with individual trajectories that lead up to the same endpoint, a neuropsychiatric disorder. If key events during brain development before birth or after birth are disturbed, that can lead to schizophrenia, autism, depression, epilepsy, etc. Henceforth, it is imperative that we create preclinical models to gain greater understanding of the developmental origins of mental illness so that early intervention will become possible. What you see here is the embryonic forebrain that looks like a coronal section, like in a coronal section. The red lattice-shaped structure is the vascular network of the embryonic forebrain. These blood vessels provide valuable guidance cues to the key events that take place during early embryonic development. If there are abnormalities in these blood vessels, it will significantly affect main events during brain development. Also, blood flow of different brain regions will be affected. The brain is made up of different cell types. The brain cells are called neuronal cells. Endothelial cells, on the other hand, are the building blocks of blood vessels. Interestingly, many genes that were believed to be enriched in neuronal cells for decades were found to be enriched in endothelial cells of embryonic forebrain in our laboratory. With the gene expression in these endothelial cells were further classified according to disease categories, and enrichment was seen in disease categories of schizophrenia, epilepsy, depression, and several other psychiatric disorders. This was a novel finding that directly linked these endothelial cells and blood vessels to mental illness. In our gene expression profile studies of embryonic forebrain endothelial cells, synaptic genes were enriched. My project involves the study of these synaptic genes which are enriched in the gene expression studies. Synapse is a structure that permits a nerve cell to pass an electrical or a chemical signal to another nerve cell. So therefore, the question arises, why the synaptic genes are expressed in forebrain endothelial cells? One of the image below you see the expression of a synaptic protein in an endothelial cell and a very curious localization. So the main scientific question that I ask here is, what is the functional significance of synaptic genes in endothelial cells and blood vessels? And what will happen when I delete these genes from the blood vessels during development? What kind of disease model will it lead up to? It will at least take me 2 years to figure this out. So this experience at HMRI will be used to enhance and develop my subject matter expertise. This will be utilized further to develop newer projects and publications. Also, a career plan will be initiated to develop funding in the future. This opportunity will be used to enhance collaborations with other researchers at HMRI to gain a diverse array of expertise in brain-heart connection, to be specific. Mental illness affects 1 billion people worldwide and costs the global economy almost $1 trillion each year. It's also a major cause of concern due to the lower rate of productivity at work. Social stigma, discrimination are factors that go unreported. With our work, early diagnosis and early intervention will become possible. Greater outreach of our scientific study will create more awareness about the origins of mental illness. This work, when implemented in developing novel therapies, will lead to better clinical outcomes and in turn improve the quality of life. My research and my findings will pave a way of giving back to the society. I hope I can make a difference. And I thank HMRI for giving me this wonderful opportunity. Thank you.
Hello, everyone. So my name is Veronica Blair Hubble, and I graduated last year from the University of Notre Dame with my PhD in chemistry. When I finished high school, I actually had no idea that I was even passionate about chemistry. It wasn't until I took undergrad organic chemistry class that I started to develop an interest. What got me interested in chemistry was learning about how medicine actually works in your body. And so it's probably known that if you have a headache, you'll take aspirin, Tylenol, ibuprofen, for example. However, when you have a stomach ache, you'll take different types of medicine. I never really thought why that was until I got to organic chemistry and started learning about how the structure of the compounds do different things in your body. And so that's what led me to looking for my PhD in chemistry. At the University of Notre Dame, I was excited to learn about antibiotic therapy, how they work, how they actually target bacterial cells and are able to eradicate infections. I was able to learn how to synthesize compounds that can help antibiotics. And so after my PhD, I was really hoping to expand on my expertise, learn how to synthesize new types of compounds, and hopefully for the idea of using it in medicine and other kinds of applications besides antibiotic therapy. And so I was excited to move to California to work with Professor Brian Stoltz at Caltech to synthesize compounds that are used here at HMRI to target and treat migraines. Sorry, one second. Get the laser pointer. So at HMRI, we're excited to learn about how to help people that suffer from migraines. It's probably not a stretch to assume that each and every one of us knows someone who suffers from migraines, perhaps you yourself. And the reason I say it's not hard to assume is because it's so prevalent. About 1 billion people within the world suffer from migraines. And because of all the side effects related to migraines, such as the photosensitivity, nausea, and don't forget the debilitating headache, it's known as the 7th most debilitating illness in the world. Even though a lot of people suffer from migraines, not a lot of people understand why migraines occur or how to treat them. And so here at HMRI, we're looking more into how does migraines actually occur in people? What's the difference in their brain chemistry versus someone who doesn't suffer from migraines? And how can we help them? Over the past 2 decades, there's a lot of research shown about what neurons or nerves are responsible for the pain related to that debilitating headache. However, not a lot known about why they deviate from people that don't have migraines. And so one thing you can take away is there's this trigeminal nerve that's known to be involved in the pain pathways associated with that migraine. At HMRI, they've shown that people that have elevated levels of sodium in their cerebral spinal fluid, which I'll talk about momentarily, actually have migraines, where people that don't have a lower level of sodium. So our hypothesis here is that when the sodium concentration is higher around this trigeminal nerve, at the same time it's excited, migraines occur. And so then in order to test this hypothesis, we want to lower the amount of sodium around this nerve and hopefully prevent migraines from even occurring. So before I get too involved in how we're going to tackle that, let's first talk about where the sodium even comes from. So each and every one of us has what's called cerebral spinal fluid, otherwise known as CSF. It's this watery substance that circulates your brain and down your spinal cord and back again. What it does is it brings nutrients to your brain and waste products away from your brain. And so some of the nutrients could be proteins, glucose, sugar, or ions such as the sodium, potassium, or calcium. However, where does this watery fluid even come from? And so there's this pink fleshy material called a choroid plexus that's part of your brain. And here on the right-hand side, it's zoomed in a little bit better. It sits in these ventricles of your brain. And its main purpose is to secrete this watery substance. And so if you look at the bottom right-hand corner, you can see it's kind of this globby 3D structure, and that's the choroid plexus. If you were to take a slice out of it and look at the inside, you'd see that there's a bunch of blood vessels that run through here, and they provide their own nutrients. What makes up the 3D structure of the choroid plexus are these epithelial cells. So if you were to zoom into this, you would see them here in the middle structure. If you zoom into one of the epithelial cells, you'll see there's a bunch of little proteins that line the outer membrane of these epithelial cells. What they do is they basically act like a gateway to allow ions either into the cell or out of the cell. And so basically, we want to try to inhibit the protein responsible for letting sodium out and therefore into this watery substance that we believe is causing migraines to occur. And so our goal then would be to basically block or plug up the protein responsible for that sodium concentration. Well, which protein are we targeting? So the protein that we're interested in is called the sodium potassium ATPase. Basically, it just means that there's sodium involved, potassium involved, and it uses ATP for energy to operate. And so it lets sodium from the inside of the cell to the outside and lets potassium from the outside to the inside. So our goal is if we can plug up this protein and inhibit that sodium from ever getting into this, the CSF fluid, then hopefully we can prevent migraines from occurring. And so where do we start? Well, thankfully for us, there's already drugs known to inhibit this particular pump. However, unfortunately, and the harder part of this project involves the fact that this particular protein is found all over your body. It's found in your kidneys, in your heart, and your brain. So how do we selectively target this particular protein in your brain and not the other tissues? And so that's where the chemistry side steps in. And so there's drugs already commercialized to target this particular sodium pump that's found in your heart cells. These are drugs such as Wabane, digoxin, digitoxygenin, and bufalin. However, these are not very brain permeable because, and you know that based on like the types of structures around the compound. However, they share a lot of similarities that we can build upon. And so the goal of my project is to use these types of compounds as a structural template to derive and build compounds that are more brain permeable and hopefully target this particular sodium pump within the brain. And so the short-term goals of my project involves synthesizing compounds that are novel and hopefully able to inhibit the sodium pump within rat brain cells. And then the long-term goal would be to then apply those to the human choroid plexus epithelial models and eventually test them in rat models as well. And so I'd just like to take a moment to say thank you to HMRI for this incredible opportunity and to the James G. Boswell Foundation for the Boswell Postdoctoral Fellowship for allowing me to be here. Thank you to my mentor at HMRI, Dr. Alfred Fonteth, and my Caltech mentor, Professor Brian Stoltz. And thank you all for your attention, and I'd like to take questions at the end.
Hi, good evening, everyone. I'm Khaja Shamim, and thank you so much for joining this presentation. Today, in this presentation, I will be talking a little bit about myself, about me, about my background, and also the kind of work that I did in the past and the work that I'm going to do here at HMRI. And also, I will be talking a little bit about the kind of research experience that I will be gaining here from HMRI, and also what kind of contribution that I will be doing, giving back to the society once I complete my postdoctoral program here from HMRI. So let's begin. Let's dive in to know a little bit about me. I'm Khaja Shamim, and I'm from India, from south of India. The hometown is Telangana. And of many things, my hometown is famous for a couple of things, and one of which is this beautiful monument, which was built in 1591. It is called Charminar, and the other flavorful and tasty delicacies that is my hometown is famous for is Hyderabadi biryani. So let's talk a little bit about my background, about my education. The desire to the passion for becoming a scientist has led to an incredible journey that led me to travel different parts of the world for science. So from India, I happened to travel to England, and from England I went to Scotland, and from Scotland I went to Sri Lanka, and from Sri Lanka to China, and ultimately here in US. So let's talk a little bit about my past research work. Apart from my PhD project, which was focused on kidney research, most of my previous work was focused on understanding cardiac-specific ATP-sensitive potassium channels and how they behave in oxygen, changes in oxygen levels, which is also termed as hypoxia. So as you can see here, this is the three-dimensional structure of this megadalton protein, and this is the two subunits, which is SUR2A and KIT6.2. These two subunits physically associate with each other to form the functional protein. And I'm talking about a cardiac-specific composition, and these changes with the tissue type. So the function of this ion channel is to, when it opens, it allows the potassium to pass through it. So the question I was working on when I was in the University of Dundee is that people, those who are living at high altitudes, tend to live longer and have healthy hearts, and the mortality is relatively low due to cardiovascular diseases in this particular population. So we wonder why it could be that these people have a tendency to live longer. So to understand this, as you can see here, that as we climb up the altitude, there is a decrease in oxygen level. So what we try is here that we try to mimic this environment in the lab and try to change the levels of oxygen and understand this phenomenon. And we, at least in parts, we believe that these ATP-sensitive potassium channels are involved in this phenomenon. So we try to understand this in the lab using changes in the levels of oxygen. So during this time period, I happened to make a couple of interesting discoveries, which eventually ended up becoming publications in international review journals. So what I will be working here at HMRI. So here at HMRI, I will be focusing on an interesting proteins, which is FLIP1 and FLIP2. And I will be trying to understand how these proteins are involved in cardiac aging and injury. So my postdoctoral supervisor is Dr. Nicole Purcell. I will be working with her to understand and address this issue and try to better understand this protein in cardiac aging and injury. So before getting into details about this protein itself, I want to show you a basic information about the cardiovascular diseases. So these cardiovascular diseases is undoubtedly a leading cause of death worldwide. Not so even in United States, as you see here, is a lot of deaths and casualty of deaths is due to cardiovascular disease, and particularly 1 in 3 deaths in US is due to cardiovascular diseases. And it is putting a lot of burden on the US economy in terms of billions of dollars are spent to tackle this problem. So if we look into the risk factors, what are the risk factors that are causing the development of cardiovascular diseases? So multiple risk factors are involved, as you can see here, but a couple of them are under control. For example, like diet or exercise that we can control. But there are some other aspects that we cannot control, for example, like aging or genetics. So there are multiple risk factors that we have to understand to actually find a better cure for cardiovascular diseases. So here at HMRI, I will be working on understanding a protein which we think that it has the potential for a therapeutic target to treat cardiovascular diseases. So what is this FLIP? So FLIP is a name that has been derived by its structure. These are of two types, which is FLIP1 and FLIP2. So the function of this FLIP is to dephosphorylate, meaning to inactivate, for example, the AKT here, and it promotes cell death and helps in tumor suppression. But over the past couple of decades, over the past couple of years, it has been found that FLIP has multiple targets, not just AKT, which is reported to be involved in tumor progression, but also it has the capacity to interact with other proteins, as you can see here, that are involved in cell survival and growth. So as this protein is very interesting that it can interact with multiple proteins in the cells and it can activate or deactivate them. So this FLIP is involved in multiple diseases. As you can see here, that it is involved in multiple diseases, and trying to understand this protein in heart not only helps us understand how this protein functions, but also the findings that we do here in the heart will also pave way to better treatment for other diseases also. So at HMRI, I will be using genetically modified knockout models. One that is global knockout, and the other one is the cardiac specific knockout model to understand two aspects of the disease. One is the aging process. So how removal of this protein affects the normal aging process, and also to understand how this removal of this protein would affect the injury process in young and aging hearts. So all these aspects will be studied using histological and biochemical studies. So now I want to talk a little bit about the kind of postdoctoral experience that I will be gaining at HMRI. So at HMRI, I will be able to acquire new technical and interpersonal skills. And also I will be gaining experience in developing novel research projects and develop writing skills that will be helpful in writing grants and fellowships. And as HMRI is generous for providing support for attending international conferences where we can present our work to scientific and non-scientific community. It will allow me to make networking and collaborations. And finally, all these aspects will help me one day to become an independent researcher. So the long-term goal and the career, the long-term goal and what I will be giving back to the community is that the work that I do here at HMRI will help in advancement of the basic research where we understand how the disease works and try to find new therapies. And ultimately, the work that I do here will pave development for new therapeutic targets. Finally, I would like to thank Dr. Nicole Purcell for her supervision and Dr. Anju for her support and for this fantastic event. And I also would like to thank HMRI for this wonderful opportunity at HMRI. And if you have any questions or you want to connect, please use my email address or you can get me in touch in Twitter. Thank you so much.
Good afternoon, everyone. My name is Chen Xia. I'm from Neuroscience Research Program. I'm a postdoc fellow. I got my PhD in neuroscience from Shanghai University in China last year. My previous research includes some brain dysfunction-related disease like tinnitus and some molecular biology-related research such as some mitochondrial genome or voltage-gated sodium channel cloning. My research here at HMRI focuses on migraine. Migraine is a very common disease. Nearly half of the people experience headache, and 12% of Americans get migraine headache. Migraine have higher prevalence in women when compared with men. The primary symptom of migraine is headache on one side of the head. Pain is sometimes described as pounding or throbbing, and it will make patients feel sensitive to light, sounds, or smells. Up to 80% of people who get migraine have a family history, but you may get migraine more often if you are high stressed. So maintain a good mood will be beneficial for your health. Migraine is not equal to headache. Based on the area and the type of the pain, there are 4 major types of headache as shows here. As we mentioned before, migraine only has pain on one side of the head, and it will concurrence with nausea, avoid light or sounds. Nowadays, triptans and CGRP receptor antagonists were the most frequently used for migraine treatment, but they may relate it with disrupt vasoconstriction or blood-brain barrier. So find some new specific target medicine for migraine become very important. Our previous research showed that the migraine patients show higher sodium concentration in the cerebrospinal fluid, which we call CSF here. The CSF here is our brain, and the CSF is a blue liquid shown in the picture. It can be collected by lumbar puncture, and it can help to diagnose disease of the central nervous system and give us more information which CT cannot tell. The choroid plexus, which Veronica mentioned before, we call it CP. It's grow in the surface of the ventricle in the brain. The ventricle is a chamber that contains the CSF inside of the brain. The CP procedure most of the CSF in the central nervous system, so we guess CP is crucial for the increased sodium in the CSF. The CP performs as the blood-CSF barrier in the brain. It has tight junctions between the cells that can forbid the solution go through it. So our first question is, where is the increased sodium come from? We can see from the picture that the CSF only have contact with the surface of the cell and the junctions between the cell. So if there's some problems with the junctions that lead to the leak from the plasma to the CSF, will it cause the increased sodium? Based on our research, we find that the sodium concentration in the plasma was lower than the CSF. That means the increased sodium was not from the plasma. And how about the cells? We can see many black dots here on the cell membrane. It's called sodium potassium ATPase. It's a transmembrane protein that can use ATP to pump the sodium outside the cell. The function of this pump was to keep the iron balance of the cell. So we guess maybe the increased sodium in the CSF was caused by the overactivity of these pumps that makes them continually pump more sodium outside the cell to the CSF. To confirm our hypothesis, we established an in vitro cell model as the picture shows here. We grew monolayer CP cell in the insert membrane. When the junction between the cell is tight enough to block the solution go through the insert membrane, it can be used for the future experiment. So we put artificial plasma inside the insert and artificial CSF outside the insert and change the sodium concentration in the CSF as we want to check the changes of the sodium pump during this period. We can also use it to mimic migraine and add different drugs in the plasma to test whether the drugs can decrease the sodium concentration or inhibit the sodium pump. Besides this, we also designed preclinical research model, as shows here. The two models are a drug-induced rat model and hereditary migraine mice model. As we mentioned before, migraine can run in families, and this mice line is derived from patients with familiar hemiplegic migraine type 2. So we plan to use different techniques to test the changes of sodium concentration in the CSF and the changes of sodium pump and junctions in the choroid plexus during migraine, and use widely used migraine treatment medicines and a specific sodium pump inhibitor called wabin to test the possibility of use sodium pump as a specific treatment target for migraine in the clinical. My previous research focused on tinnitus. It had some similarities with migraine that they were all caused by the overactivity in the brain. The reason why I'm here is that I want to experience a different research atmosphere. Since we all know America has advanced technology and research in the world, so I hope I can find my interest area by learning and communicating with my supervisor and colleagues here. And after this period, I hope I can continue my research in the brain-related disease and keep trying to find the mechanism or the new targets for some disease that's hard to treat. And whatever I've learned from here and wherever I am, I want to share my knowledge and experience with the younger generation to help them insist on their path for the research. So that's all. Thank you.
Good afternoon. I'm Divya Desai. I did my PhD in biological sciences from NMIMS University, Mumbai, India, and currently I'm working with Dr. Vasudevan in neurovascular research program. My research interests involve stem cell biology and developmental biology. So to begin with, what is mental health disorder or mental illness is any disorder which causes a person to experience different behavior and negatively affects it in terms of thinking, behaving, and feeling. And there are various kinds of mental health disorders like anxiety, bipolar disorder, obsessive compulsive disorder, PTSD, angerness, dissociative disorder, schizophrenia, and depression, amongst others. So 1 in 4 people worldwide is affected in some form of mental illness. Depression is a global challenge which affects more than 300 million people, and schizophrenia affects 23 million, as well as bipolar dissociative disorder affects almost 60 million. So current treatments, they focus on alleviating the symptoms, but there is no cure for it. So in our lab, we are trying to understand the fundamental mechanism underlying the psychiatric disorders. Abnormalities in brain cells or neurons have been extensively implicated in psychiatric disorders. But our lab's research was the first one to directly link the vascular defects that originated during the brain development to mental illness. Recent studies from our lab have identified a specialized endothelial population. Endothelial cells are nothing but a building block of blood vessels. These endothelial cell population were found in embryonic forebrain, which can contribute to psychiatric disorders. And these endothelial cells directly impact surrounding brain cell development, including angiogenesis, which is body's natural creation of new blood vessels. So in our research, we combine genetic tools with developmental biology, cellular, molecular, and fundamental biology to characterize the fundamental mechanism underlying the psychiatric disorders. In our lab, we have previously extensively characterized the mouse forebrain, and now we have recently generated human embryonic forebrain-like endothelial cells from human embryonic stem cells. The crucial part of the protocol involves use of 2 major molecules, Wnt7a and GABA, which are specifically used to generate human forebrain-like endothelial cells. And these cells, they express specifically the GABA and GABA receptor, GABA-B3. So the field of transplantation has been revolutionized by formation of induced pluripotent stem cells. Pluripotent stem cells are the cells which can generate cells from any of the 3 germ layers. So this generation of iPSC was done by game-changing discovery of Dr. Shinya Yamanaka using patient-specific or somatic cells, like be it fibroblast, be it skin cells, blood vessels, and transform into iPSCs using Yamanaka factors, OCT4, SOX2, c-MYC, and KLF4, and they can be further differentiated to give any specific cell type. So the importance or the cruciality of the cells is we can take any somatic cells, can generate iPSCs, and then it can be used to differentiate into any specific disease cell type, neural progenitors. Right now, we can even generate organoids. And the advantage of this is we can further understand the disease mechanisms. We can move into the personalized medicine, and these iPSCs, they can be further used for cell-based therapy. So the current research has already generated neurons, specifically GABAergic interneurons from embryonic stem cells. And there are endothelial cells which have been generated from human embryonic stem cells. But the iPSCs over here, during these past few months, what I have done is we have used a control human-induced pluripotent stem cells, differentiate into embryonic forebrain specific endothelial cells. These ECs, they specifically are the ones which express the forebrain specific cell type, and they were able to give expression of CD31, which is an endothelial cell marker, and GABA, which, as I mentioned, are the specific endothelial specific markers. So now these ECs, they have shown typical cobblestone morphology, express the GABA markers for forebrain cell type, and these can be then used for cell-based transplantation therapy. So now we can use the patient-specific cell type and generate the endothelial cells. So what I'm waiting for patient-specific and iPSC cell lines so as to generate the endothelial cells from these patient-specific iPSCs. So to find the fundamental mechanisms underlying the diseases in these patient-specific cell lines, obtain a corrective mechanism to repair these defective endothelial cells. So finally, I would like to thank HMRI, and I would like to thank here that before applying the basic science to applied science, we need to understand some fundamentals, some mechanisms, and it is always wonder to me that what is the reason behind why the disease is caused and how the disease is caused. So HMRI gives an opportunity to understand, to apply, and to make this research into applied field so that to get a possible therapy, and then we can apply this in the patients so as to give back the community some much needed answers. Thank you.
So thank you, fellows. That was absolutely awesome. And so now we're ready to take questions. We'd like to open it up to the audience. And please feel free to reach out to us anytime. Okay, no questions?
No, I have a question.
Okay.
This is Sandy Sharp. I couldn't figure out how to raise my hand. So the question is for Veronica. If I understood Veronica correctly, she's going to modify known blockers of the sodium potassium ATPase so that they can enter the brain more easily. But I don't understand how that targets the brain. Won't those modifications still allow those drugs to target other organs where you might not want them to be at that same level?
Yeah, sorry, I was just making sure that was the end. I didn't want to interrupt. That's a great question. So I think that's one of the biggest questions right now is, so the idea is to modify the structures enough to where hopefully, so Chin Chin was talking about how there's a blood CSF barrier, and the idea is to modify it enough. And she's actually working in the same lab as me for the neuroscience team here, and hopefully she'll, if we find something that's a really good sodium, channel inhibitor, then she'll test it further to see if it actually, can be permeable to that barrier.
Right.
And the idea is whenever, if you swallow the pill and it makes it through the circulatory system, hopefully it just will have poor enough affinity or, attraction to that pump in other organs. And whenever it finally reaches the brain, the idea is it'll be permeable enough to where it'll go through and target that pump. Does that help at all?
Well, not exactly. So but maybe I don't quite understand the physiology. So is there no concern that by providing the patient with additional blocker, that it's going to negatively affect the organs that aren't having any problems, even though it's positive, it's doing what you want it to do in the brain? It doesn't seem like are these modifications selectively targeting the brain? It doesn't sound like that's what you just said. I think you said it can also, you're expecting it to also be able to block the ATPase in other organs.
So sorry, let me reiterate. So right now, the drugs that are used for for the heart tissue, it's very selective for that particular pump. And so
okay. So you're going to try to make it deselect the heart and go to the brain.
Yes, exactly. Yeah.
Okay. Thank you.
Yeah, no, thank you.
I have a question for any of you. Congratulations on your new postdoctoral fellowships, and the tradition, especially with the Boswell Foundation, goes back over 40 years and is a great fellowship. But it's kind of interesting because you have two mentors. If you're a Boswell Fellow, one at HMRI and one at Caltech. And so for those of you who are Boswell Fellows, I'd like to know how you are finding the arrangement of having two mentors, one in each institution, and which laboratory you are spending most of the time in.
Thank you for the question. So I'm JK in the neurosciences program. So I'm technically not a Boswell Fellow. I'm funded by a different source, but I'm also having a dual mentorship between HMRI and Caltech. So I think for both of us, we are synthetic chemists. So we are making compounds currently in the earlier phase. We're spending time mostly at Caltech. But for me, so I've made some compounds and I've tested their performance here at HMRI. So I think later in the project, Veronica also will have the chance to come over and test her molecules here. So I think it's been really great because Caltech is, has a long tradition of making discoveries in fundamental chemistry perspective. But Caltech doesn't have a hospital, and I think in terms of that, HMRI is great at it's perfectly situated to take that basic science research and to translate it to clinically relevant applications. So for me, for both me and Veronica, we are attending both of the group meetings. So every week we are attending the neurosciences program research meetings, and we also have regular meetings with our mentors to give them update about the progress that we are making in our syntheses and get their feedback on some of the different things we can do. And I think Veronica has something to add.
Yeah, hey, thank you so much for the question. So when I moved here in September, I actually spent 100% of my time at Caltech in Professor Stoltz's lab synthesizing. So I have a lot of group meetings over there. But also I participate in weekly group meetings here at HMRI. And thankfully, Zoom has actually made that even easier. But being only 5 minutes apart down the road, it's really easy to come back and forth. And so I bring molecules over here for Andrea in our lab to screen for me. And she's already collaborated with, like, we've already discussed that she's going to teach me how to run the assays, just to kind of get my hands-on experience with something else. But yeah, so I find the two mentorships very exciting, and because, you know, Professor Stoltz gives me a lot of feedback on the chemical side. I even collaborate with people that are associated with his lab for how to drug design and what to think about or worry about in order to try to reduce the toxicity that's known with some of the drugs. And then I also get the whole other side of the feedback with thinking about the biological application and really all of the publication and history behind, you know, why we think that we can treat migraines. And so, yeah, thank you.
Okay, any more questions? Okay, so then thanks everyone. Thanks for participating. We hope you enjoyed this session, and we'll see you next time.
Great job, everybody. Yeah, great job.
Bye.